unfortunately won't be able to share a glass of wine and some nibbles with you afterwards. But the advantage is that we have a very large audience, and probably the largest audience that we've ever had for our annual Lane lecture. As an introduction, if I can get my slides to work. Some housekeeping. As you've noticed, you'll all be muted, and I believe all your videos have been turned off. This is just to make sure that the sound is as good a quality as possible. We will record the lecture by Cath, and it's also possible to watch it live on YouTube. We hope, and I think Cath, you've agreed that we'll be able to share the slides later on kind of our website as well. In terms of feedback, I would ask you to use the chat function in terms of asking questions. Some of you have already kind of like asked some questions in advance, but if you have any questions that you want to ask Cath, then please put them in the chat. In addition, you may also email queries to the email address that's there on the bottom, ashton@manchester.ac.uk. We are anticipating that we'll run until about 5:30 at the end. So I'm at the moment giving some introductory messages. Then we have the Lane lecture by Professor Cath Noakes, and followed by an opportunity to ask and for Cath to answer some questions. So for those of you who are not know who Ronald Lane was. So Ronald Lane was a professor in occupational medicine who was appointed as the first professor in occupational medicine at the University of Manchester in 1945, and was the longest professor up to date at the university. His tenure was until 1964, which incidentally is the year I was born. So he established a really strong department at the University of Manchester, and did a lot of work in the effects of exposures to lead, but also to effects from dust related to experienced by cotton workers, which was obviously very topical in the Manchester area. So we have had the Lane lectures for quite a long time now. We try to do them every year in memory of Ronald Lane, who unfortunately died in March 1995. So this lecture is co-hosted by the University of Manchester, the Centre for Occupational Environmental Health that I'm working in, but also by the Thomas Ashton Institute for Risk and Regulatory Research. So the Thomas Ashton Institute is a joint kind of a venture between the University of Manchester and the Health and Safety Executive, with the aim to deliver research, learning and regulatory insight to enable a better working world. And you see here a slide with the number of the themes that is covered by the Thomas Ashton Institute. If you want to know more about the Thomas Ashton Institute, then please go to its website, and you'll see quite a lot of information about the activities of this institute. So that was really what I wanted to say as part of the introduction. So I'm really pleased and honored to be able to introduce Professor Cath Noakes. She is a chartered mechanical engineer with a background in fluid dynamics, teaching and research expertise in building physics and environmental engineering, and she leads a research into ventilation, indoor air quality and infection control in the built environment. Her international recognized research group carry out experimental and modeling-based studies to explore the transport of airborne pathogens, the influence of indoor airflows, and effectiveness of engineering approaches to controlling airborne disease transmission. And not surprisingly, Professor Cath Noakes have been really kind of important and very much in the limelight in terms of the response to the UK response, scientific response to the challenges that were placed in terms of trying to control the pandemic due to COVID-19. So she was director of the Pathogen Control Engineering Research Institute from 2010 to 2014, and director of research and innovation for the School of Civil Engineering from 2014 to 2020. She's currently deputy director of Leeds Institute for Fluid Dynamics and co-director for the EPSRC Center for Doctoral Training in Fluid Dynamics. And since April 2020, she's been involved in the UK COVID response, leading a SAGE subgroup focusing on the science and underpinning environmental transmissions of COVID-19. So that for introduction. So I will stop sharing my screen now, Cath, and it's over to you. Thanks very much.
Okay, right. Let me just get my screen to share. And hopefully that works and you can see my screen. So thank you, Marty, for the kind introduction, and thank you for inviting me along to talk. I'm quite honoured to give the Lane lecture this year. So I want to talk today about respiratory disease transmission and focus on some of the complexities around how we can understand how it's transmitting. And to be quite honest, there's an awful lot that we don't know. So although we're very focused at the moment on coronavirus, actually respiratory disease proposes a big challenge internationally regardless. And if you look at some of the data from WHO, it's a few years old this now, but it hasn't substantially changed. What you can see is that communicable diseases feature 3 times in the top 10 causes of deaths worldwide, and that lower respiratory infections are number 4 on this list. And we also have tuberculosis there at number 10 on this list. So it is a very significant contribution to morbidity and mortality around the world. And I was a bit surprised when I looked at the coronavirus data. This is the worldwide data. This is where we are as of yesterday. And I think this thing that really struck me was that the number of deaths worldwide were about 1.3 million, means it's going to land in the top 10 causes of deaths for the 2020. And that's quite scary. A new disease is going to probably overtake tuberculosis, because bear in mind this data is a little bit out of date. So it is a very significant impact, and I think it has really made us wake up to the fact that we are dealing with something that thrives on human contact, and we don't fully understand not only coronavirus, but how most of the other respiratory diseases transmit. So if we think about coronavirus, what do we know so far? Well, we know that the source of this is respiratory droplets and aerosols that contain the virus. We also know that there is viral RNA in feces. It's been found in urine as well. We're not entirely sure whether this presents a transmission route though. So the evidence for infectious virus is quite limited. And we also know that actually people who are infectious have very contaminated hands, and if so, an infectious person will be, if they touch somebody, then they'll touch a surface, they will contaminate that surface. So the routes of transmission will be through the environment, those finer aerosols, larger droplets, and people touching surfaces mean that there are 3 different ways in which this virus potentially transports from one source, one person to another person. And those aerosols potentially are inhaled, causing infection. The larger droplets potentially deposit on your eyes, your nose, and your mouth. Some of those droplets will also deposit on surfaces, and those contaminated surfaces can contaminate the hands of a person who is exposed, or a fomite, something they might touch. They then touch their eyes, nose, mouth, or put something in their mouth, potential route of transmission. So we know all of this is possible. What we don't know is what the different proportions are in all of these different routes and how important these different factors are. And that makes it really quite difficult to understand how the virus is transmitting and therefore understand how we best mitigate that risk of transmission. A number of other things that we do know already from the risk factors out there. So we know that when you're in close proximity to somebody who's infected, then your risk increases. So the closer you get, the more likely you are to, I guess, come into contact with anything they emit. And that's very likely to be a higher risk when you're face to face. We do know that when you're indoors, you've got a much higher risk than when you're outdoors. There's almost no evidence for transmission outdoors, except where people are having a close face-to-face conversation. And we know that indoors, that poor ventilation is associated with higher risks of transmission, and particularly for some of the super spreading events that we've seen. Again, we know that with coronavirus and many other respiratory diseases, the risk increases the longer you spend with close to an infectious person. And obviously that's why household risks become very high, because you're spending a lot of time in close proximity. Some of the other things we've learned is that certain activities potentially increase the risk. So singing, loud speaking, both mean that you produce more droplets and aerosols. Aerobic activity, you are breathing at a higher rate and again potentially breathing out more of these droplets and aerosols. So there is evidence from some of the outbreaks that are linked to these activities where again there are super spreading events that have happened. Crowding is a well-recognised risk. A lot of that is that the more people in the environment, there's the more chance that there'll be an infector in that environment. And of course, there are more people there it can be transmitted to. We've got some data from laboratories which suggest the virus likes cool, dark and dry places. So many indoor spaces and certain settings may be higher risk than others. And then, of course, one of the real challenges with this disease is that people seem to be able to transmit it before they show any symptoms. And some people may never show any symptoms or anything that is significant. So it isn't necessary to have a cough or a fever for you to transmit the virus. And these things together all make this a really difficult disease to manage. Just to go a little bit more detail about where have we found it. So, I mean, one of the obvious things you might think is, well, let's go and look for it. So there's a raft of studies which have done some laboratory experiments on it. Laboratory experiments are particularly challenging with the live virus. It is a category 3 hazardous pathogen, so you cannot just go and do it in any old microbiology lab. You need the specialist labs, and there are so relatively few studies around the labs around the world that are able to do this. But those studies have shown that it can survive indoors, potentially on surfaces for several hours, if not several days, and that it's stable in an aerosol, again with a very low decay rate, a half-life of over an hour. And one study showed that it could survive in aerosol in a laboratory situation for 16 hours. So we know it's a pretty stable virus in the environments that we frequent. If we look at where people have looked for it, there's a lot of studies have found RNA. There are very few which have sampled infectious virus. And again, this comes down to the fact that you need a category 3 laboratory to be able to do the infectious virus analysis. So a lot of studies have just looked for RNA. Most of those studies are in hospitals. So there's a few in the public environment, but most of them are in hospital settings or close to hospitals. And they found RNA in multiple places, patient rooms, frequent touch sites, things like the keyboard and the mouse of the computer. There's a handful of studies which have sampled and published on air environments in hospitals. An awful lot of these don't find very much. And where they do find it, it's quite small. So this is a little bit of a conundrum. You know, is the fact we can't find it there mean it isn't there? Or is it down to how we sample? And the sampling means it's difficult to detect. And I think the jury's out on that as well. It's also worth noting that in the hospital settings, often you're sampling with patients who've been infected for maybe 2 weeks or more before they get into that hospital. So they may actually be less infectious by that point at all. There is one study so far that sampled virus in exhaled breath using masks. Actually, there might be 2 of those now. So there is some evidence of it in exhaled breath directly, but again, we're awaiting data to really show exactly what is in people's exhaled breaths. So, you know, we're still, even now, several months into this, there's still quite a lack of good data that shows where it is in the community and studies with live virus. I think in the community it's very hard to find, and that's not to say that just because you find it everything's safe and it's not there. It's a bit of a needle in a haystack, and I think it's very likely that you know, whereas your risk of contracting it is most of the time very, very small, if you're in the wrong place at the wrong time, suddenly your risk goes up to something very much larger. Let's go back to respiratory transmission and think a little bit more through the physics of this and the dynamics. So we've got our source person. Here's a guy, actually, I think he's spitting water, but everybody likes this image. How much virus gets emitted will depend on what's happening. So is it because they're coughing, they're sneezing, or just breathing or talking? Is it because they're undergoing some form of treatment that causes them to produce that aerosol? Whatever they produce comes with a particular size distribution. It's in a particular location, and it either happens continuously or it happens for a short duration of time. Once that virus is emitted into the air, it gets transported through the environment, and depending on the particle size, we'll get certain transport in the environment or we'll get some deposition. And then eventually people are exposed to that, and they will be exposed, as mentioned already, through inhalation, through local deposition, and through surfaces and fomites. This whole process is really complex, so it depends on the microorganism, depends on the behavior of people, and my world, which is fluid dynamics, it depends on the fluid dynamics of the aerosolization, the transport, and the exposure. And I've mentioned aerosols and droplets already, and I thought it's worth spending a little bit of time talking about what we mean by these. So these are the definitions from the dictionary, which are not particularly helpful if you're trying to understand infectious disease. But I think it's important to think about size. So the big circle on the screen is a 100 micron droplet. A human hair is about 60 microns in diameter. So that's to just give you a sense of scale here. And here is our 10 micron aerosol and a 1 micron aerosol. The virus itself, I couldn't draw on the screen, the virus is 100 nanometers, but the virus itself will never be a naked particle. It'll be contained with respiratory components, so proteins, salts, surfactants that are in respiratory fluids, and probably other bacteria and other viruses. So it'll be contained in this sort of mixture of stuff, and therefore is going to be in bigger particles. So we think we're concerned with particles that are about 1 micron in size and bigger. And in theory, the bigger the size of the particle, the more likely it is to contain virus. In practice, we're not entirely sure, because there is evidence from other diseases that and other knowledge of aerosolization of microorganisms that sometimes you can get preferential aerosolization into smaller particles than you think. But to just think about the size ranges that matter. So is it an airborne disease or a droplet disease? If we look at the classical clinical definitions of this, airborne infection refers to something that is less than 5 microns in diameter, whereas greater than 5 microns is referred to as a droplet. Now, as somebody who does fluid dynamics, I can tell you straight away that that 5 micron particle is not a droplet. It doesn't land on the surface. It will stay in the air for quite a long time. And actually, you look at the graph on the right, which I think many of you will know well from exposure sciences, actually shows what happens to different size particles when we inhale. And we can see that you can inhale particles quite easily up to about 100 microns. Most of those will deposit in the upper part of your respiratory tract. But then to get down to the deep lung, you need particles that are 5 microns, or maybe if you're lucky, not lucky, unlucky, the 10 micron particle will get down to there as well. So when we're talking about conventional airborne infection, a lot of those definitions come from, for example, tuberculosis, where the pathogen has to reach the deep lung in order to cause infection. For COVID, I don't think that is the case. There are receptors for the ACE receptors are in the nose, are in the mouth, are in various parts of the respiratory system. And therefore those larger particles up to 100 microns may matter. And of course, if we're thinking about things which land on surfaces, the bigger ones are going to land on the surfaces. So they'll be the ones that perhaps cause infections through touching a surface. It's also worth bearing in mind that you cannot see anything that's less than about 40 microns. So if you can see it, it's a droplet, it's on a surface. If you can't see it, is it a droplet or is it an aerosol? Well, we can, if you think of the sun coming through the window and you can see little particles moving in the air on a sunny day, they're bigger than 40 microns, you can see them, and they're not on a surface, they are floating in the air. So I think we need to keep a more open mind about particle size and what matters, and think through the physics of it and the physics of the exposure and make it more disease specific. To give you some idea about what people produce, this is quite old data from 1946, but it's quite nice data and it's still quite widely used. This is where measurement of particles produced by healthy volunteers and with diameter. And you can see that you get a similar size distribution across all three activities: sneezing, coughing, and counting 1 to 100 in here. The majority of those particles are around about the 10 microns diameter. You get a very small number of very large particles though too. And you see that there are vastly more particles produced when somebody sneezes than when they're just simply, when they're coughing and then when they're talking. Again, whether that matters or not will depend on what those particles are carrying. So a sneeze doesn't necessarily mean it's carrying more virus, it just means it's carrying more particles. There is a lot better data that's come out more recently which shows that actually there are higher numbers of fine particles than this older data shows. But in terms of then how much virus that carries, it might not change things that much. Sampling then for the microorganisms is much, much harder. So most of the data that exists on aerosols and droplets from people is on healthy volunteers. There are a very small number of groups who've actually measured microorganisms. So on the right is quite a simple sampler, there's a cough aerosol sampler, a CAS, that was designed to sample for tuberculosis. And really, we've known about tuberculosis being an airborne disease for many, many years, but it was only in the past 10, 15 years or so that we've actually properly sampled it from exhaled breath and got some good data on that. The one on the left-hand side is a much more sophisticated instrument. It's called the Gesundheit machine, and it's led by Professor Don Milton at the University of Maryland. And this does some size fractionation for viral aerosols and is able to determine those above and below 5 microns in diameter for people who are infected. But this is really quite a complex piece of research equipment. It's not feasible we're going to have one of these in every university around the world. Just to give you some data from Don Milton's machine, this is for influenza. And what you can see is he's sampled for coarse and fine, so below and above 5 microns. Measuring the RNA copies here, you can see that when people cough, you get an increase in the number produced. But the important thing I want to show here is the range. So you can see there is probably 4 orders of magnitude difference between the people who he's sampled in this study. And that tells you that some people who are infected are producing very little, and some people are producing enormous amounts of aerosol. And that's really quite important. There's not a clear reason why there's a difference between those people. We do know something about variation with activity. This isn't biological, this is just particles, aerosol particles measured from people doing various respiratory activities. And again, the important thing to show in here is the people breathing. there is a very big range. So just simply taking 25 people and asking them to breathe, you get a huge variation between people. You ask them to cough, again, you get a variation, but it tends to be much higher. You ask them to sing or speak, and when you get the highest loudness, you end up with the most particles, and it's fairly comparable to when somebody's coughing. Bear in mind, a cough is an instant thing. When somebody's singing, they're singing for potentially quite a long duration. And although in here you can see that the loudness when you speak and singing is very similar, really this is 90 to 100 decibels of speaking is shouting, and you wouldn't shout continuously, but you might sing continuously for quite a long time. I mentioned viral load a little bit already, and again this is actually data from very early in the pandemic. This was published in April, I think, and so it's only for symptomatic people. But what this is showing is that there is a drop-off in positive culture over the duration of the disease, and more recent data has also included asymptomatic cases in these sort of data analysis. And you can see that the highest amount of virus tends to be at the very onset of the disease. So people are likely to have the highest viral loads and therefore be producing the most virus at the very beginning of their disease. And I just wanted to touch on how much virus is in a particle. So if we are able to assume, and there is an if, that the viral load carried by a particle is directly proportional to the volume of the particles, what we can do is take some of the data on what we know about droplets that are produced and say, okay, if we assume a particular viral load, in this case it's 1 × 10⁶ virus per mil, we can actually look at the viral load carried by different particle sizes. And you can see that the very big particles in theory pack a huge punch. They carry a really big viral load. The thing is, there are a very small number of those, so there's a real lottery as to, if you are unlucky and you get hit by that big particle, then the chances are it's sufficient to cause infection, but there aren't very many of them. And I think the ones that we should be worried most about are these ones here in this sort of 10 to 100 micron range. Many of these will evaporate, so they will become more concentrated, but they're the ones which are carrying quite a high viral load. The very small ones really potentially don't carry enough, but these are the ones that could carry enough and be transported enough, and there is enough of them to potentially cause infection. But again, this is a little bit speculative at the moment. It's very hard to say that this is really what's going to happen. And I touched on their evaporation. So when these particles are released, the droplets and aerosols are released, they will start to evaporate. Now, they won't just evaporate like water to nothing. They will evaporate until they leave the non-volatile components behind. And there's a number of people who've looked at this and several calculations, and typically it's about 20 to 50% of the initial diameter. And the evaporation here happens really quickly as well. So even a 100 micron particle, that will evaporate to, say, being a 30 to 50 micron particle in about 5 seconds. So, and that's a shorter time than it takes to fall. A 20 micron particle will evaporate to its final diameter of maybe somewhere between 5 and 10 microns in less than a second. And so we can look at, there are studies which have considered this and look at how these different particles might travel and where they might get to. And what you sort of see is that the very big particles can travel quite a long way because they're ballistic. They travel a little bit like tennis balls. The smallest particles travel a long way because they evaporate, and the ones in the sort of mid-range, these are the ones which would deposit out actually quite close to the source. But it's these ones which are going to evaporate and head off around the room, which we're worried about from an aerosol perspective. A lot of the data that we can show on what happens to particles is dependent on the dynamics of the aerosol. So an awful lot of people will calculate how long does it take a particular size to settle. And that's based on the fact there's a particle settling in air, it has a drag force and a gravitational force. But we have to remember in real rooms, there's a velocity involved. And I've mentioned already the evaporation. So aerosol transport depends on what happens to the air in a room. So we can't just say, oh, settling time in still air gives us X. And to just illustrate this, this shows some simple calculations of settling time in still air. So the blue line is the time it would take to fall for particles of different diameters. So a 50 micron particle would take 30 seconds to fall 2 meters. A 20 micron particle would take 3 minutes to fall 2 meters. But we can also plot their falling velocity, so how fast they're falling in the air. And what we can see is, although this 20 micron particle in theory drops out the air in 3 minutes, it has a falling velocity of less than 0.02 meters per second. Okay, now velocities of air in a room are typically somewhere between 0.05 and 0.1. So you can really quickly see that that falling velocity is going to be less than the air speeds in the room. So that particle can very, very easily be carried and taken away and affected by the airflow. And just to show again a simple example of this, this is a computational fluid dynamics simulation, different sizes of particles to just show what happens to them. And they're all sort of ejected at a 10 meter per second in the middle of the room, as if somebody's coughed. These are the really big particles. They fly, flying spit, imagine, or don't imagine if you prefer, will go ballistically, go quite a long distance. We've got some 50 micron, 100 micron particles here. You can see that they're affected by the airflow, not very much, but they get affected. And then you can see a 10 micron particle just disappears off around the room and goes off on a track around this room and is in the air for 800 seconds here. It doesn't just fall out the air. So I think the take-home message is we need to think a lot more about this continuum of aerosols and droplets, and the classical statement of a 5 micron droplet deposits in a meter is untrue. It doesn't. Physics just say that doesn't happen. So that's just sort of thinking about the some of the physics of it. And I want to go on and think about what does this mean in terms of how we can control respiratory transmission. I'm very pleased to have worked with HSE on some of the work we've done through the SAGE EMG group, and I acknowledge Ron Macbeth who produced this diagram for us. But thinking about using a hierarchy of risk controls approach, and again many of you will be very familiar with this, and thinking about how we can use this in the context of controlling coronavirus. So in here we've got, you know, things like the obvious things, don't, you know, basically work at home and don't interact, stop an activity to eliminate and substitute these. But then I think one of the areas that gets less attention than it should do is thinking about the engineering controls. It's very easy to go to administrative stuff, cleaning regimes, putting, you know, one-way systems in place, putting PPE in place. But I think a lot more attention needs to be given to the engineering controls in here as to what we could do. And that I've mentioned screens and barriers, automatic doors, but things like ventilation is a very important step in this one too. I just also want to touch a little bit on thinking about how you might use some of these hierarchy of risk controls in thinking about prevention and mitigation. So in terms of infection, we can think about the fact that you would be exposed to a virus, so you could try and prevent how people are exposed. And this is a scenario where you might say, okay, a typical office scenario, you might interact with people in the office, a coffee bar or a meeting room. How you could apply different barriers, so elimination, you can, you know, working from home, you could have some online meetings, you can apply ventilation controls, for example, as engineering. In here, you could apply cleaning, and you could potentially apply PPE in here as well. And then we've got mitigations, which actually, if people are likely to be exposed, how do we prevent them being infected? And I think one thing that's important in here is to recognise the transmission mechanisms. So the short range droplets and aerosols when you're very close to somebody, the longer range aerosols when you're in the same room, and the surfaces, and think about how these mitigations perhaps interact with these. So ventilation, for example, might mitigate aerosols and might have some effect on short ranges, but it doesn't really do very much for surfaces. And cleaning a surface has a lot of effect on surfaces, but really doesn't do very much for the air. So this just to, a visual way of thinking through the different prevention barriers and mitigations that might take place. Now, as an engineer, I'm very much interested in the engineering control side of these things. So particularly around ventilation, how we can use different ventilation rates, air distributions, pressure controls, etc. in here, how we can use different technologies, so filters and air cleaners particularly, and how we could actually use human behaviour, so separating people out in a hospital context, scheduling treatment at different ways, and effective PPE. And I think one important factors in here is thinking about distance, which is tied into this in some way. So I've already mentioned about the different modes of transmission. When you are very close to somebody, you are inhaling this high concentration of droplets and aerosols, and we get this short range droplet and airborne transmission. When you're further away in the same room, we might have a longer range transmission that's transmitted by those aerosols. There's some further complexity in this though, some real complex physics of how these things travel. When people exhale and they're breathing, you get this plume. So the cases down the bottom show somebody breathing and somebody blowing. Actually, breathing and blowing gives you pretty much the same physics, just different velocity magnitudes. But then when you there's a very complex thing that happens when people do what's called a violent ejection, so a cough or a sneeze. The particles clump together and travel in a turbulent puff. So think about when you see somebody smoking and those particles cling together in the air for a bit longer than you think they should do. And that means that they can potentially go further than we think they do. Just to show you some of the ways in which these measures could interact, we've got the impacts of masks in here. I think this is a really nice video. It shows the flow around a person. It shows how far their cough goes. And it shows that when they put a mask on, it has a really big impact on the airflow in that space. So if you just watch her again, when she coughs, you can see just how far that cough plume goes. And if that's carrying viral particles, then it will carry a long way. When she puts the mask on, it doesn't transport as far. It goes up within her thermal plume around her body. And there is some actually quite nice data on the impact of masks on viral loads. So this is again measured with Don Milton's Gesundheit machine, and measured for a seasonal coronavirus and measured for influenza. And what you can see is that the masks are actually really quite effective at reducing both the aerosol particles and the droplet particles, and more effective actually for the seasonal coronavirus than they are for the influenza. My favourite topic is ventilation, as some of you already know. You can ventilate buildings in different ways, mechanically through ducts and fans, but also natural ventilation, either through opening windows, or the building you can see on the right is actually a naturally ventilated building with a really clever advanced natural ventilation system in it. Ventilation responds, contaminants in the air will respond to ventilation. So the graph shown here shows carbon dioxide with occupancy, actually measured in a lecture theatre. And you see that the more occupants there are, the more carbon dioxide in the air, and it changes. So we can use some of these things as a proxy, but we know that if there's virus in the air, it will respond in a similar way. And we can represent buildings with an air change rate, which is basically the flow rate divided by the room volume. And show you some examples from some studies that we did in the past. This was a study looking at using CO2 as a proxy for an aerosol exposure in a hospital setting. And we was a naturally ventilated building, which is quite a complex thing to measure in. But we were interested in the potential exposure to a CO2 tracer, which was then used to representative of an aerosol transmission. And what we looked at there was the influence of different ventilation strategies. So we've got a scenario where you've got a source, which is representing a patient here. You've got P1, 2, and 3, which are other patients, and then HWs are healthcare workers. And we've put sensors in all these locations. And what you can see is when you have the ward here is an open ward, you've got a fairly uniform distribution between each of the cases, but when you close the windows, you get about a 4-fold increase in the exposure in that setting. So it's giving us a real strong message that the ventilation in that space matters. What's quite interesting with this study is we also looked at the effect of partitions, and I know partitions are something that we've been considering quite carefully for coronavirus, mostly for large droplets. But this is showing what would happen for partitions in the case of aerosols, and we've put partitions between the beds with the same scenarios. And what you see in here, it's a bit more of a complex picture. Ignore the one which says low wind, because that was a more complex setting. But what you see is that the risks close by and to the patient directly opposite are slightly increased, but the ones to the patients to the side are decreased. So actually the message this is telling us is that a partition in a space, actually what it does to the environment depends on the ventilation flow, and it can actually increase risks for some people and decrease for others. Of course, this is just for aerosols and not for the larger droplets. One of the things we've been wrestling with is how do we actually link up the difference between ventilation in a setting and the risk. And we can use a modelling approach. So we can use something called the Wells-Riley equation. This is a way of relating infections to disease, and we use it relates it to the ventilation rate in a room, the breathing rate of people in there, and the amount of infectious doses that people would generate. So we sometimes call this quanta. It's a little bit of a catch-all parameter, and it gets back calculated from from past outbreaks, but at the same time it gives us a useful way of doing comparisons of risk between different spaces. And just to show you some of the relationships in there for what we think we know a bit about for coronavirus, we've got relationships with this is quanta, so 1 quanta per hour, 10 quanta per hour, and 100 quanta per hour is the generation rate of infectious doses versus ventilation rate under 2 different conditions, a steady state scenario and a transient scenario. And what you can see is that when you've got very low emission rate, actually the risks are quite low of an aerosol transmission, but you might get a risk only when the ventilation rates are very, very low. But when you have somebody who's a high infector, actually, even with good ventilation, it might be quite hard to control it because they're generating so much. And that's where we have to start thinking about limiting the time that people spend in those spaces, as well as improving the ventilation rate. So these relationships are quite hard to work out. We lack a lot of the data that we need to really do this in a robust way at the moment. The last thing I want to touch on for air is ultraviolet irradiation. So this has been recognised as a method which we can use to inactivate microorganisms in air. It's widely used for bacteria, and people have suggested that actually it could be effective for coronavirus. So it's basically the UV damage to the DNA of microorganisms, and it basically stops it'll inactivate the microorganisms in there. How effective it is depends on the species, it depends on climate conditions, and it depends on how much UV the microorganism is exposed to. There is a data for a human coronavirus. It's not SARS-CoV-2, but it does suggest that actually it's a relatively easy microorganism to inactivate. The data would suggest that so far. So there are different ways in which you can apply such a device. You can put one basically in a box in a room with a fan and draw the air through it, and that can either be installed, so a little bit like an air conditioning unit, or portable plug-in devices which you can move around the room. Another way of doing this is something called upper room ultraviolet air disinfection, whereby you create an open field of UVC light up above people's heads, so they can't be directly exposed to it, but then any pathogens that are in the air are carried up through that zone by the air mixing and the convection currents that are in that environment. And so we've worked on this quite a lot. We've looked at how we can model this. So this is just a model scenario where we're testing it in a room. We've got a ventilated room where the air comes in and passes through the room. And we've got these devices which sit on the walls and put a layer of light into that space. And we're able to measure the irradiation from those devices. In fact, you can see one of the devices in the picture here, and you can see you get a blue light emitted. The blue is the visible light, it's not the UV, but it allows you to see where that radiation is that's created by that light. And we've worked on modeling these. So this is a computational fluid dynamics model of the airflow within a room in this scenario, and how we can couple this in with the UV device to see where in the room would be affected. So this is showing the dose that the air in that room would receive, and this can allow us to do predictive models of how these devices behave. And what we've done is taken some of the outputs from those models and taken them into a simple zonal flow model to say, well, actually, what might this do in reality for controlling transmission? So we've got a scenario here where we've got it's a hospital scenario. You've got zones 1 to 4 are where your patients are, zones 5 and 6 are circulation zones in there, and we'd have airflow that might transfer between some of these spaces. And we assume we've put somebody infectious in zone 1, and then we say, okay, well, in the baseline scenario, the ventilation rate is 3 air changes an hour, and we haven't got any UV. In scenario 2, we've doubled the ventilation rate. Let's see what happens. But then in the other scenarios, we keep the ventilation rate the same, and we put UV devices in either the zone 1, zones 1 to 4, zones 5 and 6, or everywhere. And we were interested to say, well, if we did this, what might be the impact on infection rates, and what might be the impact on energy? And the results are quite interesting. So ignore the fact there are red and blue dots. That's just about the interzonal mixing, and it doesn't make very much difference. But here's our base scenario where we've got a fairly low energy consumption, but then the highest number of cases. If we double the ventilation rate, we basically double the energy cost, and we halve the number of cases, more than half the number of cases. But if we put the UV in our infector zone, actually we're getting close to the benefits that we'd see from doubling the ventilation rate, but the energy costs are much lower. And if we put even more UV in, we can potentially get to the point where it's even better than just in doubling the ventilation rate, but at a lower energy cost. So I would never advocate reducing your ventilation rate, but potentially if you've got poorly ventilated spaces, these types of systems might be a relatively energy efficient way of improving the environment and reducing the risk. The last thing I want to touch on is hands. So I've mainly focused on what's in the air, but I think hands matter too, and we shouldn't dismiss them. So one of my researchers, Marco, has done some great work where he's been modeling transmission via hands, considered how many germs are on surfaces. If you touch a surface, what's the efficiency of transferring something from that surface onto your hands? If you've got something on your hand, well, how efficiently can you transfer it to a surface? How much contact area you have with a surface, and how effectively and frequently you clean your hands. And we've got a stochastic model that considers this risk to be able to understand contamination on hands. We did some studies initially with biological aerosols in a chamber to be able to say, can we predict where things deposit onto surfaces? and we can manage to do that reasonably effectively. And then we did an observational study in a hospital setting where we said, okay, well, who touches what in that environment, and can we measure the frequency of those touches? So after watching about 400 sets of care, we were able to get some data which on who touches what, and this is just one example where we watched the sequence of which people touch things. It's quite interesting that you'd find that the sequence of touch, particularly in a healthcare setting, is not random. There is a definite probability that if you've touched one surface, you'll then touch another surface, which is quite an interesting observation from that. And then ultimately we're able to put these together into a risk model. So this is showing a scenario again with a hospital. We've got an infected patient in bed 1, and they're either in single rooms, and the healthcare worker goes to the patient in room 1 and then goes to room 2, goes to room 3, goes to room 4. Or we've got a scenario where we've got our infected patient in bed 1, but they're in a 4-bed room when the healthcare worker still goes 1 to 2 to 3 to 4. And what we can see in those ones is that in the single room scenario, the relative risk, the amount that's going to be on the healthcare worker's hand, drops as you go patients 1, 2, 3, 4. There's obviously a distribution in here because the distribution reflects the fact that they might not wash their hands effectively. But in the 4-bed ward, that risk doesn't decrease. In fact, if anything, it goes up slightly, and that's reflecting the fact that that environment enables the deposition of particles and the patient 1 to contaminate the environment around patient 3 and 4, for example, without them being aware of that. So we were able to put some quantification onto this. Although people say it's not rocket science that a single room is lower risk, we were able to put some quantification onto mechanisms as to why. And we can take this into modelling COVID risk. This is some really early data where we can model the decay of virus on surfaces with time based on some of the experimental data that's been published, and potentially then put this into this stochastic touch model in order to look at how many virus copies you could transfer to your nose over a period of time, depending on the amount that's on surface. So I wouldn't read too much into this, but it's a way of just showing that we can start to think about modeling those risks and considering the different parameters. And I think, you know, shows that actually the risks after a long time are going to become very, very small quite quickly. So just to finish up then, I think, what do we know so far? We still have a lot of gaps, but I think that the close range transmissions like the dominant route, that it's that mixture of droplets and aerosols. And I think it's quite likely that the main route you're going to get those is you're probably going to inhale them, even the bigger particles. That we know that the far field aerosol matters some of the time. So beyond 2 meters, if you've got poor ventilation, longer duration exposure and high aerosol production, then I think there's quite a high probability that airborne transmission may happen. And we should not dismiss the surfaces, they still matter, the contamination through deposition, but there's very little evidence that actually after a long period of time, surfaces pose a risk. I think one of the other things that we need to really consider from this is the fact that the definitions of droplets and aerosols and droplet precautions or airborne precautions, particularly in a healthcare setting, may actually be stifling what we can do, because it's a very black and white definition at the moment. And I think there's a need to update those definitions to not say we have a single cutoff at a particular size, but actually consider the fate of those aerosols and different types of aerosols and how this might change by different diseases. So it may well be a different set of different importance for coronavirus compared to for tuberculosis or for influenza, say. And I think that's something that we ought to be exploring as we go further forward once we've got over the immediate crisis with this. And practical recommendations, from everything I've seen so far, I would say assume that aerosols do matter in transmission. It may still be precautionary in many cases, but I think we need to assume that they do. I think I would say that you should be identifying poorly ventilated rooms, trying to mitigate those and reduce their use. I think it's really important now that we look at how we maintain ventilation through winter. And then thinking about if we've got a poorly ventilated space, don't put the air conditioning unit on, try not to position fans so it disperses that air. And I think it's quite important that we do now start to consider whether air cleaning devices may become an important part of our mitigation in places we can't improve the ventilation. And I would also say don't ignore any of the transmission routes. So I think we don't have enough information yet to assume that we can dismiss things. I think we will over time, we'll start to narrow it down, but for now we need to put that proper hierarchy of risk controls in place and a layered approach that addresses all of the possible transmission routes. And to finish up, there are a lot of people to say thank you. A lot of people's work featured in here, so particularly the people mentioned on there, but also to everybody who I've worked with at Leeds, collaborators, and particularly want to flag the people within the SAGE Environmental Modelling Group who we came together in April and we've worked together solidly. Since there, we've produced a phenomenal amount of output, and there's an awful lot of people who've contributed a huge amount of time that feed into our knowledge of COVID transmission so far and hopefully help keep us safer. So with that, I'm going to finish and take any questions. Thank you.
Thank you, Cath, for an excellent presentation. It's really kind of outstanding. I think the way that you pulled it together and managed to summarize the large amount of complexities that we're dealing with in this in relation to the transmission risk and control. So thank you very much. I found it a really kind of useful, interesting, and I learned a lot from what you've been saying in the last 40-45 minutes. There's been quite a lot of comments on and questions on the chat functions, and we also received some questions beforehand. So I suggest that I'll start with and you've seen those already. So I suggest that we start with those.
Okay, can you read them out? Because I haven't got
yeah, of course I can. Let me just. So the first question that we had from Joe Bowen was: it's clear that the UK public has got the message that it's beneficial and also necessary to wear face coverings in public indoor spaces or when 2 meter distance cannot be observed. It's also clear the message about how to wear face coverings correctly has not reached the UK public. In what ways could this communication gap be filled?
Yeah, I agree, and it is a difficult communication gap because I think a lot of the communications, it's about how do you message to different audiences. So but I do agree there is a gap in that, and some people who are possibly willfully wearing them incorrectly. I think some people are just aren't aware that they're wearing them incorrectly. I do also think that actually as we go further forward, it might be quite important to try and figure out if we can get a standard for face coverings so people know if they're buying one, is it going to be of any use or not, and how we could measure that.
Thank you, Cath. Then the second question that was submitted beforehand was about offshore environments. So would an environment like offshore where people are in an enclosed space with common HVAC systems aid in transmission of a respiratory virus such as COVID, even if people maintain social distancing where possible?
So if you've got an HVAC system, potentially it could transmit it. There is as yet very little evidence that the virus is transmitted, sorry about that, within through an HVAC system, so between two rooms. It seems that if there is aerosol transmission, it's within the same space at the moment. The only, I suppose, caveat to that is there are certain settings and possibly chilled environments which are highly refrigerated, use a lot of recirculated air in order to keep the environment cold enough. They may be a case where it is more likely that there's a risk within the HVAC system.
Thank you. And a question related to Christmas. How will we be able to safely sing carols this Christmas, standing on our own doorsteps 5 to 10 meters apart? Question mark.
Yeah, so carol singing at Christmas, that I'm aware that's a live issue at the moment. It's a tricky one. So singing is, unfortunately, it's a real shame that it is being shown to be one of the riskier activities. And, you know, it is because it's such a social thing for people. But at the moment it is. I think if people were going to sing, I think outdoors is definitely far safer than indoors. Still maintaining distance, possibly don't need to be as far as 10 meters, but you know, 2 or 3 meters outdoors definitely, and possibly wearing face coverings. I know it's not the most ideal thing, but if you know, some of the singing is not about giving out the best performance, it's about joining together in a community.
That certainly would be the case if I was singing, I think. The next question is from Christopher McCarthy. How good are P2 masks at reducing transmissions? So are they actually better than other face coverings, I guess?
Yeah, I mean, so face covering does two things. It acts as a source control, and that's predominantly why they're being advocated. So if you have the virus and you don't know you've got the virus, it is actually preventing you from sending that out into the environment. And actually, even the cloth face coverings are quite effective there, because often it's those bigger droplets that we're trying to catch, and even the smaller aerosols start life a bit bigger. So if you can catch them in a face covering before they evaporate, then it can mitigate that. As a protective, so the FFP2, FFP3 are all about a protection, and that's why they have that the marker on them. So yes, they would provide more protection against aerosols. Most of the time, in most environments, we probably don't need that level of protection. It's quite interesting starting to watch the data come out on the face coverings, though, in that some cloth face coverings perform, I wouldn't say quite as well as an FFP2, but they're performing, getting close to it if they're well fitting.
Okay, and sometimes you see these masks with a little vent in them. What would you say about wearing?
Yeah, so the problem with the little vent is that, okay, it might still provide some protection to you, but it doesn't provide any protection to anybody else. So as a source control, it's not very good. You're just letting your coronavirus out into a nice jet for your friends to breathe.
So nice directional kind of.
Yeah,
like
nice
that,
directional. Don't stand downwind of a valve mask.
Right. Okay. There's a question from Neil Dans. He says, I was interested if there are ways of disrupting the spread of aerosols in public transport design, so in the future planes and trains can reduce the risk of future transmissions of all disease. Now, I've also seen some comments from Ashley Woodcock who says that actually the risk on planes appears to be extremely low. But
yeah, so planes on the whole is quite low, because from an aerosol perspective anyway, because the ventilation rates are really high on planes and they already have very good filtration in their systems. Trains is a bit more of a lottery. So strangely, the London Underground is actually very well ventilated. It's got a very high airflow rate, and I think your risks on the London Underground are going to be much more to do with if you're close to somebody. But other trains, buses, it's really hard to say what their ventilation is. And I wouldn't want to make a judgment. A lot of us can say, oh, that's poorly ventilated, but is it? We're not actually sure. We also have to remember that ventilation will only mitigate a bounced aerosol transmission. So although we think that's an important route, it's not the only route that we need to consider. So we need to make sure that we're also considering that close range transmission and the surfaces as well.
Okay, thank you. Another question in relation to tobacco smoke. Does tobacco smoke transmit the virus further or hold it in the air for longer?
I'm not aware of any evidence that it does. So I couldn't say for sure, but I don't think so. There's certainly no evidence of transmission via smoke. Interestingly, there is some associations with poor air quality and COVID risk, but that seems to be that it's people in those areas are likely to be more susceptible, as opposed to the air pollution itself is a carrier.
Okay, then the next question, I don't think there was a name attached, is the use of HEPA filters, is that practical in your view?
Is it practical? It really depends on your setting. So within recirculating HVAC systems in buildings, I don't think it's practical or even probably necessary to put HEPA filters into most of those systems. Most of them are not designed to take them. I think it's a good idea to make sure that whatever filters are in there are in good condition and are kept in good condition. And it's also a good idea to limit the amount of recirculating air as best that you can in those systems. There is discussion about using portable HEPA devices or other filtration devices that I'm not very up on my filter grades, but have a fairly high filter grade. And again, they may work in certain settings, but you have to be really to put an air cleaning type device in a space. You need to really think through who's using that space, when are they using that space, what's the size of the space, what's the flow rate of the device, what's the noise that the device produces. If it's a portable one, is it just going to get tucked away in the corner or kicked over? So there's a lot of factors. You know, there's no magic bullets with this, unfortunately.
And that's interesting. You know, all these things need to be taken into account when designing interventions, I guess. Next question was from Bryony Ross, who I think does exercise classes, provides exercise classes, and her question is about how ventilation could be used to enable safe, closer proximity exercise in indoor environments such as gyms or exercise and dance studios. Given that, as I understand it, if people exhale much more forcibly when exercising, and two, that mask wearing is not regarded as recommended for higher levels of exertion.
Yeah, so exercise classes is difficult, and it's difficult to say getting closer than 2 meters, because even with good ventilation, it won't disrupt the environment beyond about 1.5 to 2 meters. When you're in that very close proximity, it's that immediate high concentration that matters. If you've got people who are more distant, absolutely ventilation matters in those settings, and particularly the high aerobic type activities. There have been a couple of outbreaks reported. There was one in South Korea, which was quite interesting because it was an aerobics type dance class where the transmission happened, but the same teacher who was infected also did a yoga class, and nobody in the yoga class was infected, which might suggest that the high intensity in that setting was a higher risk. There was also one in Canada which was a spin class, and again they'd applied everything, they'd got the distancing, they weren't wearing masks, but there was still transmission that happened.
Would air curtains be helpful, both in terms of cooling down the person who's exercising and trying to prevent transmission?
Interesting idea. I don't know. I think it would be more complex. That I think that's perhaps a longer term investigation type answer.
Yeah. Then another question from Husan Lin. Can a closed room with only one window ventilate naturally effectively?
So it can. Window spaces ventilate better when there is more than one opening. So often in your house, I mean, I find this, I've got sash windows and you open one and it doesn't do much. So then I have to go and open one in another room across the back of the house to make it ventilate. But yeah, you can get ventilation. The more openings, the better. If you've got one of those windows that allows you to have a top opening and a bottom opening, that's actually far better than just opening a single point in a room. But this time of year, once it gets colder, ventilation is driven by two things. It's driven by wind and it's driven by temperature. So the bigger the temperature difference, the more ventilation you get. So actually you can get almost as much ventilation from a fairly small window opening in the winter as you would from a wide open window in the summer.
Interesting. I wasn't necessarily aware of that, so that's an interesting fact. I think we probably addressed the issue about the airplanes and why the risk is lower. You kind of said there's a combination probably of ventilation and filtration. Ashley suggested perhaps also seat configuration.
Yeah, absolutely. I think looking at, particularly proximity to people and trying to keep people further apart, sitting people in family groups, and wearing masks.
And the next question is about masks. How would anyone know how effective the graphene mask filter is? I believe University of Manchester provided their students with graphene based masks.
I have no idea. Maybe the University of Manchester will measure it. Okay, I haven't seen the data on the graphene masks. So.
Okay, thank you. How true is it, Catherine Gaffney, how true is it that the quicker the airflow in an aircon, the less risk there is on passing on virus?
So I guess it's the thing that matters most is how much outdoor air you get in. So the more outdoor air, the lower the risk of the virus building up in the space. Air conditioning units are quite difficult things to deal with, because some air conditioning units are connected to a ventilation system and bring in fresh air, so they can be quite good. Others simply recirculate the air in the space to cool it, and how much risk that poses is likely to depend on the environment that you're in. So if you're in a very poorly ventilated environment, they may pose a risk because they are continually circulating the virus. If you're in actually quite a well ventilated environment, they probably don't pose very much risk, because although they're circulating the virus, it's such a small amount compared to how much you're getting from the dilution from the ventilation that kind of overrules it. And so I would say, if you've got an air conditioner, if you're confident your room's well ventilated, by all means use it to keep yourself comfortable. But if you're not confident, then think twice about using it.
Thank you very much. The next two questions I'll take together. It's about the use of UV as a risk mitigation measure. Simon Clark suggests, could UV also increase mutation rate of the virus? And Hilda Palmer kind of asked whether whether UV systems might create ozone, and is there a trade-off between well, is there not a trade-off, are there potentially consequences that perhaps are not foreseen by applying some of these risk mitigation strategies?
Okay, so on the mutation, I think it's very unlikely the UV will cause mutation with a virus. It's possible it does for bacteria, because with a bacteria you get DNA damage and then you can get a photo repair mechanism. And so I don't think anybody's actually proven that you get mutation, but it's possible. I'm not a microbiologist. I'm sure there are people in this room who know more than I do about this, but I don't think you get that mutation, because I don't think you can get a photo repair with a virus. I think once it's damaged it, it's damaged it. In terms of ozone generation, some UV systems do produce ozone, some don't, and I think the well-designed ones that use the right lamps don't produce ozone. It's all to do with which wavelengths of UV it produces, and certain wavelengths potentially can create ozone. I would never advocate for a system that generates ozone. There are a number of air cleaning devices out there that use all sorts of different technologies. Many of them use ionizers, plasma devices, all sorts of things out there. A lot of them have a byproduct, some sort of chemical byproduct, which is I wouldn't want to advocate for those, because I think there's a risk that you've just swapped one harm for a different harm.
Okay. there's still a number of questions. Are you still okay to continue?
Yeah, I can answer a few more. Yeah.
Okay, that's great. So another question from Neil Dans, who asks, would the cost-benefit analysis of ventilation and UV technology against all communicable diseases justify having these everywhere?
That's a really interesting question. I think that sounds like a research project. I mean, we looked at it in a very simplistic way for one scenario. I think you would have to look at the benefits for different diseases. And of course, you know, one thing that's quite interesting about the mitigations that we've seen so far is that in schools particularly, coronavirus is spreading, but it's not spreading like wildfire like you might expect to see it, like you do with many other diseases. But kids went back to school and all of them got a cold within a week. Yeah. So it would suggest that whatever we're doing to control COVID isn't necessarily working to stop common cold. Or maybe the infectious dose for that is just so low, I don't know.
Excellent. Then a question from Johnny Taylor. Thanks for a very interesting talk. Do you have any practical advice on how to manage ventilation with open windows? How do you know what the ventilation rate of a window is? How do you keep the room warm? Anecdotally, I know school teachers who are struggling to follow advice as it's too cold and are not sure what to do. Yeah,
so that one is a tricky one. Especially rooms which are naturally ventilated, it's very, very hard to measure. So there's two things you can do. One is that if you're in a room with enough people, you can measure carbon dioxide. It won't give you a perfect measure, but it's a proxy measurement. And if your carbon dioxide is, say, and if it's above 1,500 parts per million, you can be fairly confident that your room is not well enough ventilated and you need to do something about it. If your carbon dioxide is below about 800 parts per million, you can be fairly confident the room's reasonably well ventilated and it's okay. So that's one thing you can do. If you've got one of those meters, you can use it to sort of gauge when you open the windows and when you don't. If you do use that, you need to buy one that's got a non-dispersive infrared, an NDIR sensor on it, because some of the cheaper ones don't measure carbon dioxide. What they do is measure VOCs and then do a calculation and estimate it. Right. In terms of practical, how do you manage it? So think about risk. So if you're in your house by yourself, or just you and your family, you're all interacting together. So actually having your windows open all the time is probably not a really good plan. So you want to open windows when those risks are likely to happen. Now schools, again, are a more difficult setting, but again, I think what you can do is not necessarily have them all wide open all the time. You don't need that much air. You can open them a little bit. I'd always say have some background ventilation, have them cracked open a little bit all the time if you can, but then open them maybe for 10 minutes every hour. To air the room, and then that means you're not freezing quite so much, but you're allowing that virus to disperse out. And of course, if you can actually match that up with a break, so if you can get outside and kick all the kids out in the playground for 10 minutes, it then helps you air that room even better. So it is a difficult one, though. I realise that for people, and I think this disease has thrown a spotlight on things like ventilation and how we design our buildings. We've designed our infrastructure focusing on things other than health.
And I think that's a very pertinent point. Actually, there's a question a little bit further down from Hilda Palmer again. The best CO2 meter for cost.
Yeah, I'm not very good at the actual. I mean, I think you need to think about how you're going to use it, who's going to use it. So there are some quite good ones, but they're really techy, and so they don't necessarily work for everybody. The really cheap ones often say don't measure CO2 directly, so you have to, I mean, they will still give you an indication of really poor ventilation and very good ventilation, but you have to be careful about not reading too much into the data, and you should never read too much into the data anyway. Just because your CO2 spikes a little bit once or twice doesn't mean it's poorly ventilated. So really thinking about what you're watching is part of it.
So a question from Ashley Woodcock. If you cough on hand, what proportion sticks to the hand versus propelled as droplets?
Yeah, I don't know that, but I do think that this is an underestimated thing, that I think people are very fixated on virus coming out of somebody's mouth and depositing gently onto surfaces, but actually coughing onto somebody's hand, or wiping your nose with your hand, wiping your mouth with your hand, I do think that is a potential route that we are not really aware of. And the problem with that route is it's very stochastic. It's, you know, you get either nothing or lots of something. There's very little sort of in the middle.
Thank you. Then actually another question about CO2 meters. Can we use technology, CO2 meters, to provide real-time indicators for good and poor ventilation in healthcare or teaching spaces as a use, don't use indicator for room occupants?
So yeah, you could do. Of course, your CO2 only works when the room is occupied. So it measures exhaled breath. So, you know, if you put it in a room with nobody in, it won't work. It doesn't tell you anything. And I think one of the things in there is about, it's not just what you've got to learn. What do you do? How do you respond to those CO2? And what does it mean for people? It's quite interesting that I think there's a real piece of research needs doing around how people understand ventilation. And I forget, because I live for ventilation. So, you know, I forget that people don't necessarily understand it. And, you know, it's been a surprise to me, I suppose, that people don't know what a trickle vent is, or didn't know that they had a ventilation system in their building. And I guess that's what I've just had to learn through this. But yeah, there's a piece of work about how we understand these things and how we understand how our buildings work. There's education in there.
And hopefully after all of this, people will know a lot more about ventilation than
I
before
hope so.
before
I never thought I'd have the Prime Minister say ventilation.
The next question, again from Ashley Woodcock, is could it be in the cold food chain, presumably the virus, and could that be a transmission route?
So there is some suggestion. There's been the odd suggestion from the media that it's been found on frozen food packaging moving between countries. I think it is true that it will survive in cold environments. I think if it was in the chilled food chain and being distributed through the supermarkets, we'd have a lot more random transmission than we've got at the moment. So I don't think that's a major route, but I think definitely chilled food processing is an environment, and meat processing are environments where we know there's high risk of transmission. How much of that is to do with the environment and how much of it's to do with workplace interactions and social interactions, shared cars, people living together, there's all sorts of different things in there to unpick.
Next question from Andrew Curran. The performance of any ventilation system will be affected by a wide range of factors in addition to the physics. This might include the approach to the maintenance of equipment, age and performance of the kit, the ability of individuals to affect the systems, etc. Can the models take account of these additional risk factors?
So in theory, yes. I mean, we haven't done, but in theory, yes, you can. I mean, you'd have to figure out exactly how to characterize them, but you could put them in as probability distributions into models to take account for the fact that you've got inefficiency. And some of the models that we run would naturally account for that anyway. So, for example, we've done some fairly simple analysis looking at air cleaning devices. And all of those, we've basically said, let's assume it's only delivering 50% of what it should do, to take account for the fact that it is likely to give you an incomplete mixing in that environment.
The next comment, I'm not entirely sure, it's probably in relation to something that you mentioned during the presentation, but it's from Hilda Palmer, and she already kind of admits that it's possibly political. So I'm not sure whether that means whether you have an excuse not to answer it, but you said, why, and I'm not sure whether it's related, but why is this not coming through from you, from SAGE, EMG and HSE in terms of workplaces, most of which are very poorly ventilated and overcrowded, especially schools and offices? So I don't know what this refers to.
So yeah, I guess, I mean, I'm an independent member of SAGE as a scientist, so my role is to give advice, and of course I do research in this area, and I do a certain amount of media discussion in this area, but it's not for me to make policy decisions, unfortunately. So if I was making policy decisions, I might have got ventilation on the table earlier, but it's not my policy to make. And I think, you know, some of it is not so much about policy, it's about how we get messaging to people. It is a bit difficult to, and we need to figure out different ways of getting that messaging across to people. I would also say that we have learned as we've gone through. So some of the information that comes through, it sounds like we've only just come up with it, but actually it's you know, it was inappropriate to put certain things on the table until you've got some evidence for it. So we, you know, things do change, and we learn more and we update things based on what we've learned.
Brilliant, thank you. A question from Alex Mayares, if I pronounce it correctly. Would you say that it's justified to request the public to use face coverings outdoors as well, which countries like Spain are enforcing?
So I think I would say on a risk-based approach. I think if you've got people in close proximity outdoors, then it would be appropriate to recommend it. If people are well distant, then it's the impacts are likely to be really quite small. So there's so little evidence of transmission happening outdoors that it would be an unnecessary thing in most circumstances.
Okay, next question again from it says here, Ashley Woodcock. So data on mask contamination, taking off masks, contaminating hands, reuse, etc. Should they be single use? Should masks be single use?
I think again, it depends on the risk and the environment that you're in. So if you're in healthcare settings where your chances of coming into contact with the virus are much higher, then probably so. But if for public settings, our chances of coming into contact with the virus are still quite small, and the chances of you know, if you think about you've been in an environment with somebody who's infected, so you've been breathing in the air, they've worn a face covering, you've worn a face covering, maybe they haven't, you've worn a face covering, it's protected you, so but your face covering is now contaminated. It's going to be quite difficult to then say, okay, I take that off, I get enough of it on my hands, I then touch my nose to get infected. The chances are that that's not going to happen. It's quite a small risk. And so I think as long as when you take your mask off, you're a little bit careful with it, you keep your hands clean and try not to touch your eyes and your nose. And then I think washing them regularly. And I think the bigger risk there is not so much the coronavirus. I think a lot of that is the fact that masks are going to get pretty nasty if you wear them for too long, and so washing them regularly. But I worry about single use from a sustainability perspective, because there's an awful lot of plastic out there if we're not careful.
Okay, I think we're not going to be able to get through all the questions, so perhaps two more questions, if that's okay with you, Cath. So yeah. There's a question from Martin Regan. Are some settings more of a priority for innovative ventilation solutions? I guess are more settings more liable for super spreading events than others?
Yeah, I mean, I would say every setting should be looking at their ventilation, and most settings can do something about it. You know, your first step is actually to understand how is the fresh air coming into your space. But I think if we were thinking about innovation, the settings where people come together, hospitality settings, schools, some of the workplace settings, those environments where the transmission is likely to happen more readily than others, perhaps where you would be interested in looking at these things. And I think, you know, there are technologies out there. There are some good technologies, there are some bad technologies. I think it would be a good time to start to say perhaps we should be looking at trialing some of these technologies and seeing whether we can actually get some good data on them.
Okay. And then I think the last question that I think we can really kind of ask for you, because you've given already so much of your time, and I know that how busy you are. I think we kind of addressed the question about cloth mask and exhalation valve mask. So there's a question from Delphine Bard, and could you help dentist surgeries being able to perform necessary drilling activities?
Yeah, so dentist surgery, that's been a really difficult one, because we know that you've got an environment there where somebody is unmasked. It's a higher risk environment, and the fact that you're doing drilling, that you're potentially aerosolizing the virus. So yeah, I think that is a setting where I think the focus on ventilation in those settings, more full stop, has been forgotten about for many, many years. And people are saying, well, my dental surgery doesn't have any ventilation, and I would possibly take the view that, well, if it doesn't have any ventilation, it's contravening all the building regulations and you shouldn't be using it. But I think we do need to start thinking about how we can innovate in those settings. Now, they are one of the settings where potentially use of air cleaning scrubber type devices could be used. I think we need to think quite carefully about how they're used, and we need some data on those. And it's quite difficult to get that data and get good data on that. But again, thinking about how we could support dental practices in improving ventilation in their premises.
Yeah, and I think I'm aware of some work some collaborative work between, I think it's York, University of Manchester, and Cambridge, where they designed some, I mentioned again, these kind of air curtain type control measures to trying to protect the dentist, really. Okay, I think we need to kind of bring it to the close, because it's gone past 5:30. Cath, thank you very, very much for an extraordinary kind of like excellent presentation, but also being able to authoritatively answer all these kind of wide range of questions. I think it's been really enlightening, and I think it was really kind of good to have you, and thank you very much for giving up your time to talk to us and deliver this Lane lecture. It's been excellent. So thank you very much, and thank you very much everyone else for your interest and joining this Lane lecture. And providing the questions, and there's many more questions in the chat, and I'm sure that and if we had the opportunity to have a glass of wine and some nibbles, we could have really kind of extended some of these discussions quite happily, I guess. But unfortunately, I can't give you a virtual glass of wine this time. So
I'll have to go and get my own, will I?
Yeah, I should have done that. I should have sent you one over the post, Cath. I apologize for my my lack of foresight.
I'll come back to next year's lecture when you're back in reality, and I'll have my glass of wine then.
Okay, well, I'll get you two then. Okay, well, thanks very much. Thanks very much, Cath. Thanks very much, everyone. And enjoy the rest of your day and the rest of your evening.
Thanks a lot.
Bye-bye.